What happened before the Cambrian explosion? Today, we are setting out to explore a little-known era, a forgotten frontier between ice and life. Imagine a world where life, still in its infancy, is slowly redefining the rules of living beings. A world without trees, without fish, without shells, but already bubbling with biological experiments. This is the Ediacaran, a fascinating, pivotal time where everything suddenly accelerates. More than 600 million years ago, after emerging from the most extreme glaciations in Earth's history, life no longer just survives. It unfolds, organizes, takes root in the ocean floors. Microbial mats cover the shallow oceans like vast gelatinous forests, fueling an explosion of strange forms, sometimes fractal, sometimes immobile, but already of unexpected complexity. This is where it all begins. Oxygen rises, the first nerve networks appear, and mobility becomes an evolutionary weapon. It's here, long before dinosaurs, long before trilobites, that life begins its grand narrative, one that will eventually lead to us. But before embarking on a new adventure, don't forget to like the video and subscribe to the channel to make sure you don't miss anything. Thank you, and have a nice trip. Here we are at the dawn of the Ediacaran, between 635 million and 600 million years ago. This stage, both distant and fascinating, marks the beginning of the Ediacaran, a geological era named after the Ediacara Hills in Australia, where fossils of some of the oldest known multicellular life forms have been found. But before understanding the importance of these life forms, we must closely examine the geological and climatic context that shaped this pivotal chapter of Earth's history. The Marinoan glaciations, named after the region of Merino in South Australia, were so intense that much of the globe may have been covered in ice. Experts believe that at the height of these episodes, the planet's average temperature dropped well below the values we know today, with surface waters sometimes just a few degrees Celsius. Over time, the gradual increase in temperatures caused the melting of the massive ice caps, releasing oceanic zones previously trapped in ice. The meltwaters, loaded with sediments and various ions, then altered the chemical composition of the seas, promoting the stabilization of the ocean floors. Before this, the unstable and cold environment left little room for life's diversification. Now, a less harsh marine world was emerging, offering organisms the first refuges to settle and develop. In this renewed setting, shallow seas saturated with carbonates appeared. To better understand this phenomenon, let's remember that carbonates form when carbonate ions bind with calcium or magnesium ions within seawater. This chemical bond occurs more intensely in warm conditions, where the water chemistry has already been disrupted by the melting of ice. The resulting rocks are primarily dolomites, grouped under the name of cap carbonates. The word cap expresses the idea of a layer placed over older formations, thus signaling the end of glacial ages and the entrance into a more life-friendly environment. These dolomites are significant geological indicators. They are found on many continents, proving that the transformation of the oceanic landscape occurred in a more or less global manner. The thick accumulations of carbonate sediments reveal a major upheaval in the geochemical cycle, linked to the planetary shift happening at the time. The term international basal marker often describes these cap carbonates as they correspond to the geological boundary between the frozen world of the past and the blossoming of a new age. Geologists frequently use sudden changes in sediment or rock chemistry to determine where a period in Earth's history begins and ends. Thus, finding a layer of clear, homogeneous dolomite extending from Africa to Australia and across other continents allows us to define relatively precisely the beginning of the Ediacaran. Beyond this role as a temporal marker, These rocks also tell the story of how rising sea levels and the melting of ice caps created shallow, sunlit coastal areas rich in nutrients. These conditions provided life with a new playground, vastly different from the much more hostile environment that prevailed during the preceding glacial period. The Ediacaran fossils are often thought to represent the oldest known forms of multicellular life. However, long before this period, red algae already existed around 1.2 billion years ago, and even older structures, like the Gabonionta, date back 2.1 billion years. Nevertheless, no direct lineage has been established between these organisms and the forms of life from the Ediacaran. In these renewed seas, scientists describe a microbe soup, referring to the abundance of microorganisms and the density of microscopic life floating or carpeting the ocean floors. The dominant entities were then true bacterial communities, often called microbial mats. A microbial mat forms when a multitude of bacteria and sometimes microscopic algae develop in successive layers, secreting a slimy matrix that traps sediments. Since there were virtually no burrowing or sediment-mixing organisms to disturb them, these mats could spread unhindered. One can imagine entire surfaces covered in a colorful gelatinous felt, capable of photosynthesis and complex chemical transformations. Since photosynthesis is performed by capturing carbon dioxide and light, these mats could fix a large amount of carbon and contribute to generating oxygen. They thus constituted an essential energy base for organisms capable of exploiting these organic deposits. One of the fundamental features of this period is the absence of bioturbation, that is, the stirring of soil or sediment by burrowing animals. Without worms, crustaceans, or other mobile creatures to displace or overturn the mud, the seafloor remained relatively intact. Consequently, the microbial mats remained cohesive and continued to accumulate, sometimes forming raised structures. This situation favored the creation of specialized ecological niches. Certain microalgae or bacteria lived on the surface, feeding on light, while others, deeper down, exploited already produced organic compounds. The communities thus formed could sustain themselves over vast areas, neither disturbed nor consumed by potential predators. This largely explains the dominance of microorganisms during the first half of the Ediacaran. Such living tapestries represent a crucial step in the broader context of evolution, as they paved the way for the colonization of the sea floor by future, more complex eukaryotic beings. Let us fast forward in time to between 600 million and 575 million years ago. Specialists call this era the Avalon Assembly, in reference to certain geological regions where astonishing fossils have been discovered. During this distant time, Earth was undergoing a decisive phase in its history. After extreme climatic events and profound environmental transformations, it witnessed the emergence of a new generation of living organisms in the shallow seas. These marine beings presented uncommon forms, sometimes difficult to classify within the categories we know today. To understand them, one must dive into a scientific narrative that combines discovery, curiosity, and wonder. These organisms are grouped under a name that might surprise: Charnia, Rangea, and Fractifusus. These creatures, which were a major part of the Avalon fauna, all share a morphology described as fractal or radial. To fully grasp the idea of fractal morphology, let's imagine a pattern that repeats at different scales. When we look at a fern, for example, we can notice that each branch resembles a miniature version of the entire plant. Scientists use the word fractal to describe this kind of repetition of shapes, which is also observable with Carnia and Rangea. If you enlarge or shrink certain segments of their anatomy, similar branching patterns are found. In the case of a radial morphology, you might think of a disc or circular pattern with several axes of symmetry, somewhat like a wheel or a cake, cut into identical slices. The combination of these two types of structure, fractal and radial, gives the organisms of the Avalon assembly an unusual appearance that continues to intrigue paleontologists today. Charnia, the most well-known of the group, often appears as a large, flattened, and elongated leaf anchored to the ocean floor. Its name refers to the location of its first discovery, Charnwood Forest in England. At the time Charnia lived, there were no large animals freely swimming in the oceans, nor true predators as we imagine them. Charnia could thus grow in rows or dense colonies without fear of being devoured. Its borders, organized in successive branchings, gave each individual a delicately segmented appearance. Rangea, for its part, also exhibits a fan or frond-like segmented configuration, with repeated divisions emanating from a central axis. Finally, Fractifusus is distinguished by a more fusiform shape, sometimes described as an elongated spindle, also featuring internal repetitions of structures. All these living beings primarily deployed vertically, rising slightly above the ocean floor to capture available energy and nutrients. Why did these organisms choose to organize according to these fractal and radial patterns? A commonly advanced explanation is based on the optimization of resource absorption in an environment particularly rich in microorganisms present on the surface of the sediments. The sea at that time, relatively shallow, was covered with microbial mats. To clarify what is called a microbial mat, imagine a thin gelatinous coating formed by myriads of bacteria and other microscopic organisms that clump together on the sediment surface. These mats, sometimes referred to as biofilms, are a prime source of food and energy for any creature capable of drawing nutrients directly through its body surface. The fractal passive creatures like Charnia, Rangea, and Fractifusus had developed a way to anchor themselves above these biofilms and use the mechanism of osmotic absorption. Osmotic absorption relies on the phenomenon of osmosis, that is, the movement of a solvent, here seawater, through a semi-permeable membrane. In the case of immobile marine organisms, this means they allow water and the nutrients it contains to pass through their tissues without moving. However, this feeding strategy requires a sufficiently large body surface to maximize collection. Consequently, the fractal configuration of these creatures increased their surface area for exchange with the surrounding water. The more extended the surface, the faster and more efficiently nutrient absorption takes place. In calm seas, where competition for food was essentially vertical, that is, rising higher than one's neighbor on the sediment, these anatomical devices provided a considerable advantage. Scientists have also discovered concrete evidence of these feeding mechanisms in laminated sulfur films, that is, successive layers of sulfur-rich sediments where intact organic remains are sometimes found. Within these sediments, the fossilized imprints reveal the existence of entire colonies affixed, even sealed in place. As we move forward through this period ranging from 600 to 575 million years ago, we observe an increasing diversity of forms. This means that in the sedimentary rocks, we identify other kinds of organisms, which are still related by their general appearance and lack of mobility. Specialists sometimes refer to these ecosystems as static gardens, because these environments contained a wide variety of living beings that, for the most part, remained stuck in the same place and did not seem to move. Paleontologists are constantly looking for traces of crawling or walking in the rock layers, because when an organism moves on a soft substrate, it almost always leaves a trail, a stripe, or a slight indentation. However, in the Avalon assemblage, the near total absence of these mobility imprints indicates a silent world where life develops through surface colonization rather than active movement. This massive immobility is not solely a sign of limbo or weakness. On the contrary, it was probably an adaptive choice, dictated by the ambient conditions. Fractal organisms could enjoy an easy life, suspended above their microbial carpet, without having to hunt or flee. The fact that they remained planted throughout their existence did not prevent them from reproducing. Scientists today hypothesize that reproduction could occur by emitting propagules, miniature versions of themselves, which would then attach not far from the original colony, thus perpetuating the occupation of the sediment. This slow expansion, favored by the absence of major external threats, allowed these creatures to cover large surfaces. To better understand the massive presence of this fauna at a precise moment, it is useful to look at the example of Mistaken Point in Newfoundland, Canada. This place has become famous among paleontologists due to its exceptional preservation of Ediacaran age fossils, including those of the Avalon assemblage. Mistaken Point consists of cliffs battered by the Atlantic Ocean, where time and erosion have exposed ancient sedimentary layers. In these layers, more than 10,000 scattered specimens are recorded, but with the peculiarity of being almost all perfectly preserved and still in place. Why such a concentration in situ? The simplest explanation, supported by many clues, is that a volcanic flow or a deposit of pyroclastic ash would have suddenly covered the seabed in a few moments, trapping and fossilizing all organisms in a protective sheath. This phenomenon is comparable to what happened in Pompeii, much later in human history, when the eruption of Vesuvius froze the Roman inhabitants in their daily activities. Here, at Mistaken Point, we are talking about marine organisms frozen just as suddenly, perhaps within the span of a minute, by volcanic material deposited underwater. Therefore, geologists later named this site the class photo of its time, like a timeless snapshot showing hundreds of Charnia and Rangea grouped together, along with other typical species from the Avalon assemblage. Each fossil remained in the exact position it occupied on the seabed, sometimes with its vertical alignment preserved, its anatomical details intact, and the complete configuration of the colony around it. What makes this class photo even more fascinating is the precision with which it allows us to perceive the spatial organization of these communities. Paleontologists can analyze the distance between two individuals, compare the relative dimensions of the fronds, or spot repetitive patterns within the same fossil patch. Here, there is no trace of furrows that would suggest the passage of a crawling animal. Everything seems perfectly stationary. The majority of these fossils are elongated or fan-shaped, organized like tufts of feathers. You can even distinguish the minute folds in their fractal structures, proof that they were not merely vague stranded jellyfish, but indeed living organisms in the open water, attached to the sediments and feeding directly from the surrounding water flow. Regarding climatic conditions, although we do not have an exact measure of water temperatures from that era, some estimates based on geochemical analyses suggest that, in shallow areas, the water could have reached values close to 20 degrees Celsius. These temperate waters, combined with low food competition and a moderate but sufficient amount of oxygen, could have created habitats conducive to the establishment of large immobile populations. Section 3. Between 575 million years and 560 million years ago, a fascinating phenomenon occurred in the shallow seas and coastal areas of our planet. The seabeds, still largely dominated by vast expanses of microbes, began a crucial transition. This phase is often called the carpet revolution, as biofilms, meaning groups of microorganisms capable of organizing into thin sheets, began to develop into truly thick and complex structures. When the word carpet is used, it refers to a thin, organic layer, comparable to a very flexible kind of felt. However, during this period, these very carpets became significantly thicker and more structured, an evolution that is anything but trivial. The microbial communities, composed of bacteria, archaea, and other microorganisms, accumulate and secrete sticky substances known as extracellular polymeric substances. These substances form a mesh that is both flexible and resistant, capable of trapping suspended sediment particles. Hence, the carpets no longer just stick to the bottom, they become more voluminous, even rise slightly, and gradually form small undulations. These low reliefs create, in a way, micro-landscapes made of hollows and bumps on the ocean floor. This organization, while modest at first glance, proves to be critical for the future. Scientists sometimes use the term microtopographic relief to describe these minute variations in height, from a few millimeters to a few centimeters, that dot the surface of the biofilms. Specifically, these are small channels and folds that alter how water circulates over these colonies. Water no longer flows uniformly as on a flat floor. It enters the hollows, bypasses the bumps, and redistributes oxygen, nutrients, and mineral particles differently. This redistribution gives the microbial carpets a stratified profile, offering terraced niches, where parts of the carpet receive more light, and others get more decaying organic matter. The play of shadow and light, along with a differentiated nutrient supply, creates an incredibly varied environment, even on the scale of a few square centimeters. In these environments, a multitude of organisms, mostly microscopic, adapt to these variations. The sediment particles retained by the extracellular polymeric substances become the framework onto which other microbes graft, thereby forming an even larger and stronger platform. As the microbial layer thickens, it fosters the verticalization of organisms, a phenomenon suggesting that some life forms increasingly strive to stand erect to capture light and exploit these new niches. Beyond this reshaping of the biofilm landscape, another major transformation is underway. Fossils found in certain regions, like those attributed to the genus Fractofusus, reveal characteristics potentially corresponding to sophisticated reproductive strategies. Fractofusus is an enigmatic organism endowed with an elongated, oval, or sometimes more complex shape, which is often found in large numbers on fossil surfaces. When studying Fractofusus assemblages, one notices that individuals exist in several sizes, presumably reflecting different growth stages. All these growth stages are grouped in the same geographical area, indicating a common origin, or at least a coordinated mode of propagation. Why do scientists talk about early indications of probable sexual reproduction? Sexual reproduction generally involves the fusion of distinct reproductive cells, possibly from different individuals, to form an embryo which then develops into a new organism. Multiform animals that master this process can quickly diversify and colonize new areas. In the case of Fractofusus, mathematical models and statistical analyses have led to the conclusion that the spatial distribution and variety of sizes observed in the fossils could not be explained solely by simple clonal division or local budding. On the contrary, these data suggest the existence of a larval dispersal phase, i.e., the release of small dispersing forms that settle farther away. Researchers have thus developed scenarios where, after fertilization, tiny cells would deposit on the microbial mat, grow in place, and eventually form new colonies. This hypothesis, though still debated, challenges the idea that these large pre-Cambrian organisms were merely passive or condemned to asexual multiplication. It perhaps attests to a reproductive innovation that allowed some species to sustain and effectively spread in their environment. In addition to these considerations about reproductive biology, another element proves crucial for understanding the revolution of the mats. The local chemical changes, specifically the appearance of oxygen gradients in coastal mats. Oxygen is an essential gas for many organisms, particularly those that breathe aerobically, i.e., with oxygen. However, at that time, the overall oxygen content in the oceans was not yet as high as it is today. Scientists estimate that the concentration of dissolved oxygen varied significantly depending on the region and the depth. Parts in direct contact with the surface could contain enough oxygen to support moderately active life, but in deeper areas or confined zones, oxygen was much scarcer. Microbial mats, as they develop, create a mosaic of microenvironments. A few millimeters below the surface of the mat, conditions change radically. Light decreases sharply, the availability of oxygen plummets, and communities of microorganisms are different. Additionally, chain chemical reactions, notably the decomposition of organic matter by bacteria, lead to the production or consumption of various compounds. To detect the appearance of the first oxygen gradients, geologists and paleontologists rely on the analysis of oxidized pyrite in sediments associated with the mats. Pyrite is an iron and sulfur-based mineral, sometimes called fool's gold for its gold-like appearance. When it is slightly modified on the surface, this suggests exposure to oxygen, as this mineral is stable only in reducing conditions, i.e., with little or no oxygen. The deposits of oxidized pyrite found in layers dating from this period indicate that fine pockets of oxygen existed within the microbial mats, enough to locally transform the pyrite. These thin oxygen gradients are of major importance as they open new habitats for forms of life capable of tolerating or even seeking higher concentrations of oxygen. Indeed, as the microbial layer structures itself, one can imagine organisms taking advantage of oxygen-enriched micropockets to thrive. Perhaps some adapted to exploit these oxygen bubbles, moving through tiny openings in the mat in search of more favorable conditions. It is not excluded that this chemical differentiation supported a boom in diversity in still little-known groups by providing them a more varied and energy-rich environment, since aerobic respiration is significantly more efficient than modes of respiration without oxygen, called fermentation or anaerobic respiration. In addition, the interaction between sunlight, microbial photosynthesis, and oxygen supply creates a closely linked cycle. Through photosynthesis, certain cyanobacteria produce oxygen from carbon dioxide and water using light. Then, this oxygen diffuses into the mat, dissolves in the water, and sometimes ends up being used by other organisms to break down organic matter. This synergy improves the overall efficiency of the ecosystem. The mats, therefore, are no longer mere passive coverings, but true ecosystem engines. Of course, there were still vast portions of the ocean where the bottom remained poor in oxygen and dominated by layers of muddy sediments. However, locally, in certain coastal waters, these thick and structured mats already foretold more complex ecosystems, where the chemistry of oxygen acted as a key lever for evolution. In parallel, this undeniable thickening of biofilms and the existence of oxygen pockets induce another phenomenon: sediment trapping and the stabilization of the seabed. The extracellular polymeric substances form a kind of glue that is particularly effective. When sediment particles, sand grains, silt, remains of microscopic algae, or volcanic debris settle on the surface of the mat, many find themselves trapped in this sticky matrix. Waves, oscillations, or currents can no longer carry them away as easily. The result, the seabed gains instability. This increased consolidation creates a firmer substrate, allowing other organisms, possibly larger or heavier, to settle. They attach to it or slide across the surface without immediately sinking into unstable mud. This new development leads to a snowball effect because the more the mat retains sediment, the thicker it becomes, the more it can host other forms of life, generating in turn greater complexity in biological interactions. Gradually, the mats become the pillars of true microreefs, or, at the very least, small marine mounds, highly valued by future organisms in search of new niches. Nevertheless, it should not be imagined that everything was happening idyllically and uniformly. These mats, although expanding, had to contend with the challenges of their environment, such as possible fluctuations in sea level, the massive arrival of volcanic particles after an eruption, or even variations in salinity due to freshwater inputs. Large storms or tsunamis could, in a single sweep, clear and tear these microbial surfaces over kilometers before they slowly reformed. Besides these occasional disturbances, other more gradual factors also contributed to the environment's dynamics. The gradual changes in global climate, modifications in oceanic chemistry, and the potential evolution of certain bacterial communities. Under the influence of biochemical reactions, sediment trapping, and localized oxygen production, a subtle state of equilibrium was being built, sometimes broken, then renewed. Section 4. Between 560 million years ago and 550 million years ago, a very particular window of our terrestrial history opened in what is called the White Sea assemblage. This interval of the Ediacaran, which immediately precedes the famous Cambrian, saw a significant transformation in the seabeds of that time. In the sedimentary layers that have preserved these distant witnesses, today's researchers discover astonishing marks, betraying the progressive appearance of movement in primitive organisms. For the first time, life no longer simply stretches out in static mats or floats passively. It takes action, interacts with its environment, and leaves behind a series of traces that we are now able to interpret. At the beginning of this period, the seabed surface in the White Sea region, which corresponds to areas now located near northern Russia, although the continents have obviously drifted since, was not yet roamed by large creatures with solid skeletons. On the contrary, the landscape was dominated by microbial mats, that is, thin layers composed of microorganisms like bacteria and microscopic algae. These mats played a fundamental role. They served as a substrate, a nourishing environment, and perhaps even a temporary refuge for beings in search of food. The marine storms, less intense than the cyclones we know today, could sometimes stir the waters. According to geological and paleoclimatic reconstructions, surface temperatures, likely situated around 20 degrees Celsius, allowed for a relative stability of the ecosystem. The organisms that occupied this environment had every reason to exploit these vast microbial layers, a true natural pantry rich in organic matter. It is in this context of seabeds covered with a thriving microscopic life that one begins to observe, in certain rocky outcrops, physical traces of movement. Paleontologists, by carefully studying the layers, have identified slight notches or sinuosities that traverse the fossilized surfaces, sometimes parallel to each other, sometimes intertwined in an entanglement that testifies to a real biological ballet. Among these remarkable signs are the traces attributed to genera like Dickinsonia and Yorgia. Dickinsonia resembles an oval organism, subdivided into segments arranged in a fan shape, while Yorgia shows a similar morphology, though more elongated and sometimes featuring different patterns on the body's surface. Scientists have long debated the nature of these creatures. Were they stranded jellyfish, giant fungi, or even distant cousins of worms? Recent studies tend to view them as enigmatic forms, potentially related to the earliest animals biased toward a bilaterally symmetrical organization, that is, a body presenting two symmetrical halves, left and right. Why is a crawling movement attributed to these life forms? The answer lies in the distribution and shape of imprints associated with the trails they leave. Indeed, paleontologists sometimes observe what looks like scrape marks, as if the organism gently moved over the microbial mat surface in search of nutrients. The feeding lines detected around certain imprints suggest that Dickinsonia and Yorgia fed directly on the mat by scraping or absorbing the organic matter. By comparing the width of the grooves, their direction, and the remains of the surrounding mat, one comes to the hypothesis that these soft-bodied animals moved slowly and left regular marks of their movements. Although rudimentary, these clues constitute one of the oldest pieces of evidence for voluntary locomotion oriented towards food in the fossil record. During this same time frame, another creature draws attention, Cambrian, often described as a probable scraping Lophotrichozoan. At first glance, the term Lophotrichozoan may seem puzzling. It is a large group of animals that includes, among others, mollusks, annelids, that is, segmented worms, and brachiopods. They are called Lophotrichozoans because many of these creatures possess, at a certain stage of development, a crown of small tentacles used for feeding, called a lopophore, or go through a planktonic larval stage known as a trochophore. Today, we know that Cambrian probably had an anatomy reminiscent of a primitive mollusk. Scientists have found the presence of fine striations on certain fossilized areas that might correspond to fossilized radular scrapes. The radula, in the world of modern mollusks, is an oral structure equipped with a sort of grater or toothed organ. It is generally used to scrape food, such as microalgae covering coastal rocks or nutrient particles dispersed over the substrate. Its presence in Cambrian suggests that the animal was equipped, from that era, with a specialized tool for feeding, enabling more effective scraping of microbial mats and extraction of the organic matter that served as its energy source. The emergence of Cambrian, and by extension that of other soft-bodied bilaterans, indicates an important step in the formation of a more complex trophic chain. Until then, the concept of the food chain relied mainly on the production of organic matter by microorganisms and the passive absorption of this food by stationary organisms. With the arrival of these proto-mollusk grazers, competition for food intensifies and more sophisticated feeding strategies emerge. The interaction between microbial mats and these new herbivores is perceptible in the fossil records. On several surfaces, scientists have noted alignments of parallel micro-scratches, described as scarifications, which seem to have been caused by the repeated passage of an organism equipped with a rasping organ. This phenomenon is sometimes referred to as mat herbivory, by analogy with the behavior of modern grazers, like certain marine gastropods, capable of scraping rocks to extract microscopic algae. This transformation paves the way for a complexification of the trophic chain. Organisms no longer merely lie down or stand upright without moving. They begin to physically interact with their environment, moving sediments, damaging the microbial surface, and thus creating new ecological niches. Through these comings and goings, the surface layers of the mat are transformed, altering access to light for photosynthetic microorganisms or causing the release of buried nutrients. New equilibria are established as these first grazers expand their reach. Scientists consider this a decisive transition. The appearance of mobile creatures, even if their speed is probably very slow, intensifies the flow of matter and energy at the heart of the ecosystem. This diversity of feeding strategies contributes to the creation of producer-consumer type relationships, a key concept of any modern food chain. In the wake of these discoveries, another fossil has sparked passionate debates: Plexus racii. Initially, many thought it was simply a burrow or tunnel left by a digging organism, perhaps a primitive shrimp or a discrete worm, that would have traced this furrow while feeding on the sediment. However, a detailed reevaluation published in 2025 completely revisited this interpretation. Successive analyses showed that Plexus racii actually represents a real tubular organism, a primitive giant flatworm, and not just a corridor carved into the mud. Scientists conducted comparisons between certain preserved segments and recognized structures in modern flatworms. They highlighted similarities in the arrangement of soft tissues, which clearly directed the classification of Plexus racii towards the family of bilateran animals, lacking an internal or external skeleton. The discovery is fundamental because it implies that a great variety of shapes that did not benefit from mineralized parts could exist, while also reminding us of the difficulty in retracing an evolutionary history based on creatures primarily composed of soft tissues. The fact that Plexus racii is now interpreted as a primitive giant worm also brings insights into the potential size of organisms. Until this confirmation, biologists often imagined small flatworms moving little or simply living under microbial mats. Yet, the reevaluation of these fossils highlights dimensions far larger than those of modern flatworms, up to 80 centimeters or 30-inch long, suggesting the possibility of an ecosystem where even soft-bodied organisms could reach surprising proportions for the time. It can be assumed that the abundance of available nutrients, combined with the relative stability of environmental conditions, favored the growth and diversification of the fauna. Geochemical measurements taken from the sediments of the White Sea indeed indicate a gradual increase in dissolved oxygen, albeit limited. This rise, even modest, could have been sufficient to support the lives of these larger flatworms, allowing them an active enough metabolism to move and feed regularly. The study of Plexus racii and other primitive animals from the White Sea assemblage also helps to better understand the variety of evolutionary adaptations underway. Far from being strictly confined to the surface, some of these organisms must have exhibited more varied lifestyles. Some could still fix themselves passively, while others began to search for food by moving around. This broadening of behaviors reflects an ecosystem on the verge of advancing to a higher stage where the distribution of resources conditions the emergence of new survival strategies. The more mobile an organism becomes, the more it can escape direct competition on a fixed portion of microbial mat, and the more it opens itself up to various food sources. In parallel, this creates a stirring of sediment, promoting its reoxygenation and the release of nutrients, which maintains an increasingly dynamic ecological cycle. This revolution in locomotion, if it can be called that, is not limited to the traces left by the organism. It transforms the entire structure of communities by creating unprecedented interactions. For example, evidence of mat herbivory has significant ecological implications. By scraping or eroding the surface of biofilms, these animals pave the way for ecological succession. After the passage of a scraper, the surface can be more easily colonized by certain opportunistic microbes, while others disappear due to the lack of a stable substrate. A popular idea among scientists is that this process of moderate disturbance promotes biodiversity by making the environment more heterogeneous. This dynamic anticipates what occurs in more recent ecosystems, where herbivorous species or burrowing animals maintain, sometimes inadvertently, a mosaic of mini habitats conducive to the establishment of multiple different communities. Section 5. The interval extending from about 550 million years ago to 538 million years ago corresponds to what specialists call the Nama assemblage. This is a pivotal period marked by the emergence of life forms capable of creating mineral structures, the formation of small tubular reefs, and the gradual modification of microorganism communities. It should be noted that early biomineralization is probably the most notable element of this period. Biomineralization is the process by which a living organism synthesizes rigid or semi-rigid structures from minerals present in its environment. In the case of the Nama assemblage, this phenomenon is notably illustrated by two emblematic fossil genera, Clodina and Namacalathus. The genus Clodina comprises tubular creatures that produce small tubes of calcium carbonate, i.e., calcareous structures. The genus Namacalathus, on the other hand, appeared in the form of tiny baskets or strange cups with a mineralized wall. In both cases, these organisms lived attached to the marine substrate, i.e., fixed to the seafloor or other solid surfaces surrounded by water, such as shells or already established microbial mats. Calcium carbonate, a mineral salt consisting of calcium, is the essential raw material for constructing these tubes or shells. Within Clodina, one can observe cylinders that interlock with each other. Each tubular segment is continuously produced by the creature, probably to protect the soft body inside. For Namacalathus, the shell is less elongated, but it has a circular opening and relatively thin walls, approximately a few tens of micrometers thick. The micrometer, as a reminder, is a unit of length equivalent to 1/1000th of a millimeter, allowing for the description of dimensions smaller than what the human eye can discern. The thinness of these shells or tubes perfectly illustrates the spirit of biological experimentation that prevailed at the time. The manufacture of a mineralized protection required drawing from seawater the calcium and carbon that would form this envelope. Biomineralization strengthened the resistance of these organisms against various environmental factors. Indeed, a calcareous structure offers better protection than a body without such armor. With these tubes or shells, organisms could defend themselves against ocean currents, suspended particles that might damage or bury them, as well as against early predators. Even though predation was not yet as developed as it would be in later times, there is evidence indicating that some animals had already begun to feed at the expense of other creatures. The Clodina formations thus contain traces of circular holes, suggesting that a predator literally drilled through the wall to reach the organism inside and feed on its soft tissues. This type of fossil evidence is called a predatory drill hole. It is a perforation or hole in the shell linked to the action of an organism specifically attacking its prey to extract its food. This gradual intensification of predation, even if still modest, contributed to the emergence of a local explosion of thin skeletons. In other words, several species began experimenting with innovations in the composition and shape of their mineralized structures. In a shallow sea, rich in calcium and carbon ions, organisms could easily synthesize calcium carbonate to arm themselves. This competition for survival likely encouraged the evolution of new architectures of tubes, shells, and small tubular reefs. Paleontologists sometimes use the term adaptive radiation to describe these trends. Each new environmental pressure pushes certain species to specialize or intensify their defense and nutrition mechanisms. Ecologically speaking, what's also remarkable in the Nama assemblage is the emergence of a true three-dimensional organization. During earlier eras, many organisms remained spread out on the seafloor or lived integrated in microbial mats. With the multiplication of tubes and calcareous structures, real habitats with relief began to appear, consisting of small tubular reefs. When we talk about reefs, we often think of modern corals, which build large edifices that can house a wealth of biodiversity. In the Nama assemblage times, these nascent reefs were, of course, more modest, but the principle remained comparable: mineralized organisms agglomerated to form a substrate higher than the seafloor. Other species, called epibionts, then attached themselves to the external wall of these tubes. An epibiont refers to a living being that grows on the surface of another organism, often without seriously harming the host. In this context, an epibiont could benefit from better exposure to nutrient-rich currents and, consequently, gain easier access to its food. Within these tubular constructions associated with Claudina or Namacalathus, the spatial arrangement allowed for several species to coexist on vertical and horizontal surfaces. This leads us towards a nascent three-dimensional ecosystem, where competition is not just about spreading on the floor, but colonizing various heights or interstitial spaces. The gaps between the tubes could serve as refuges for certain microscopic life forms, or slightly larger but still soft organisms seeking shelter from currents or small predators. Over time, the accumulation of these tubular conglomerations created new local microlandscapes for the surrounding fauna. In this environment, the security provided by the mineral structures played a crucial role in the species' success. In times of danger or unfavorable conditions, these rigid envelopes could mean the difference between survival and extinction. By carefully studying the fossils from the layers of the Nama assemblage, paleontologists are able to reconstruct this mosaic of lifestyles. The tubular shells have reached us in the form of more or less well-preserved fragments. Sometimes these fragments assemble, creating an ancient miniature reef that has transformed into rock over millions of years. Geochemical analyses performed on these fossils allow the measurement of the content of various elements like calcium, carbon, and magnesium in the calcite, a mineral based on calcium carbonate, which constitutes the majority of them. Occasionally, traces of rarer elements are also found, revealing the degree of mineralization or local variations in seawater composition. These data indicate to researchers that, during these intervals of time, the availability of ions necessary for skeletal formation was stable enough to allow the development of multiple mineralized species. The multiplication of skeletal trials, with more or less thick tubes or more or less closed shells, demonstrates that natural selection was already operating on a notable scale. Individuals with slightly stronger walls, or capable of quickly repairing a perforated tube, had a better chance of reaching maturity and reproducing. In turn, their descendants could gradually improve this shell to better defend against potential predators. This evolutionary escalation, still in its infancy at the end of the Ediacaran, would later become a powerful driver of diversification during the Cambrian. However, it is striking to note that not all organisms of the Nama assemblage became good candidates for mineralization. Some species remained shellless, relying instead on their ability to hide in sediment or attach themselves to existing supports. Thus, a fairly varied palette of adaptations is discerned, from protective shells to stealthy lifestyles, to the quest for ecological micro-niches in rocky crevices or interstices of tubes. It is also during this phase that the first steps towards three-dimensional ecosystem complexity are consolidated. The microreefs of Cloudina, for example, could serve as anchor points for small algae or colonies of microbes, favoring the capture of light if the water was clear enough. Even though no fossils attest to complex algae comparable to those known today, it is highly probable that primitive marine plants exploited these environments to carry out photosynthesis and thus locally enrich the resources in dissolved oxygen. Marine oxygen was still unevenly distributed depending on depth, ocean circulation, and the density of biofilms. However, a general trend towards an increase in oxygen in the seas is noted during the Ediacaran, which helps explain why some more mobile animals are beginning to appear in the fossil record. The verticalization of habitats and the ability to access better oxygenated areas create new opportunities for fauna to move over short distances. Researchers believe that the emergence of complex interactions, such as predation and symbiosis, already heralds the diversification that will explode later during the Cambrian. Although large-jawed predators or creatures with robust pincers have not yet been found, every small circular borehole evidenced on Cloudina is a valuable indication of this dynamic. Similarly, microtraces of bioturbation, even if they seem discrete, prove the existence of organisms active enough to explore the sediment and perhaps search for food there. This exploration, in the top few centimeters of the sea floor, represents a major advancement. It indicates that life no longer merely exploits the surface of microbial mats passively, but is beginning to invade the underlying layers. Section 6. During the Ediacaran period, three major elements deeply marked the evolution of life: the progressive increase of dissolved oxygen in the oceans, the first appearance of rudimentary nervous systems in some organisms, and the influence of astronomical cycles on evolutionary dynamics. These combined factors contributed to shaping an environment conducive to biological diversification and the emergence of increasingly complex life forms. During most of the Precambrian, the available oxygen levels in the water remained relatively low, which in turn limited the complexity of marine life forms. Recent geochemical measurements, obtained notably through the study of sulfur and iron isotopes trapped in ancient sediments, suggest an increase of more than 40% in oxygen concentration in some tropical areas, particularly on shallow plateaus. To understand what these percentages mean, remember that oxygen is produced by photosynthetic organisms, especially algae and cyanobacteria, which release this gas by absorbing carbon dioxide. Thanks to this increasing input, marine habitats became more conducive to the development of multicellular organisms, allowing nascent animal lineages to grow in size and diversity. In these environments, mats composed mainly of bacteria and microalgae are capable of releasing enough oxygen into the water to support new types of organisms. Suddenly, larger creatures requiring a higher metabolism can appear and colonize these spaces. These microbial mats, at the heart of primary production in those distant times, play a driving role in the emergence of animals with more elaborate tissues. This is a fundamental change because for an organism to develop complex organs, it must have an adequate supply of oxygen necessary for energy production at the cellular level. Researchers believe that this oxygen jump, even though it remains modest compared to our modern oceans, was sufficient to propel evolution toward more diversity and the almost simultaneous appearance of mobile life forms and more varied feeding behaviors. Alongside this phenomenon, we observe the emergence of primitive nervous networks in several groups of Ediacaran organisms. Before this period, most multicellular life forms did not exhibit complex nervous structures, relying instead on passive diffusion of chemical signals across their tissues. However, discoveries of fossil traces suggest that over time, some animals began to exhibit more organized behavior, indicating the existence of a nervous circuit, albeit rudimentary. Paleontologists rely notably on the observation of sinuous tracks in the rock, where interrupted paths by abrupt changes of direction are seen. This type of mark is interpreted as a reaction to an external stimulus, such as the detection of a chemical disturbance or a change in light. Thus, the development of these early nervous networks likely allowed new organisms to interact better with their environment, adapt their mode of movement, and eventually conquer new ecological niches. To understand the significance of this innovation, it is important to first remember that any form of nervous network, even very simple, provides the organism that benefits from it with a significant evolutionary advantage. Indeed, the ability to perceive its environment, respond to stimuli, and develop mobility strategies ensures better food search, potential defense against predators, and finer internal regulation. In the Ediacaran context, where new life forms begin to move to feed on microbial biofilms or small organic particles, the presence of a nervous network, even elementary, enables more precise positioning on the substrate and an immediate response to dissolved oxygen variation. This initial stage of nervous coordination can appear in several forms. Some organisms may have a diffuse system of nerve cells along their body wall, while others already possess a slightly more pronounced concentration of these cells, outlining the beginnings of an information processing center. Studying the fossils from the end of the Ediacaran period, scientists also noticed that these early nervous networks had to work in synergy with the organism's physiology to manage the distribution of oxygen and nutrients. In a rapidly changing environment characterized by higher oxygen levels, such an adaptation becomes crucial to make the best use of available resources. Some fossils show weak evidence of body segmentation, suggesting a more advanced anatomical organization in which internal ducts could facilitate the delivery of oxygen to the most active tissues. Although these structures remain rudimentary compared to those of Cambrian fauna, they indicate functional diversification. This dynamic is illustrated by a gradual increase in the average size of organisms and the emergence of fossil traces indicating a greater variety of movement behaviors, such as superficial burrowing or smooth sliding on limestone and sandy surfaces. At the same time, another factor influenced the evolution of life during this period: the astronomical cycles described by Serbian mathematician Milutin Milankovic. These cycles, now known as Milankovic cycles, are based on periodic variations in the Earth's position relative to the Sun. They include changes in orbital eccentricity, axial tilt, and precession, which is the wobble that the Earth describes on its axis. During the Ediacaran, these cyclical fluctuations would have had a direct impact on climate, surface water temperature, and nutrient distribution. Recent studies even suggest that every 405,000 years or so, environmental upheavals occurred that were significant enough to influence evolutionary dynamics. During these phases, the local increase or decrease in sea level, combined with temperature changes in degrees Celsius, would have favored the alternation between periods of rapid diversification and episodes of localized extinction within Ediacaran communities. In these intervals of upheaval, microbial mats and diversifying organisms would have experienced varying selective pressures. For example, a lowering of the sea level in certain areas could temporarily expose depositional zones to open air, causing the demise of populations adapted to a stable aquatic environment. In contrast, in other basins where the water level suddenly rose, an enriched supply of nutrients from sediment mixing could stimulate evolutionary surges. This alternation of favorable and unfavorable phases, partly dictated by Milankovitch cycles, would have acted as a driving force in the geographical distribution and specialization of Ediacaran species. Thus, groups capable of tolerating a wider salinity gradient or quickly adapting to temperature fluctuations had better chances of expanding and thriving. Conversely, more specialized life forms dependent on a very stable environment could disappear locally, giving way to more resilient species. According to some hypotheses, Milankovitch cycles may also have influenced the global chemistry of the oceans by modulating the input of freshwater, sediments, and nutrients. Wetter periods, due to a different distribution of precipitation zones, could, for example, bring more organic matter and minerals via rivers, thereby nourishing coastal microbial mats and stimulating peaks in oxygen production. Conversely, drier periods or a contraction of wet zones could reduce these inputs, forcing microbial communities and adjacent organisms to resort to more modest survival strategies. Over millions of years, this cyclical orchestration would have shaped a complex biogeographic landscape, where Ediacaran biodiversity varied both according to local conditions, dissolved oxygen level, temperature, salinity, and following the rhythm imposed by astronomical variations. Geological records preserve chemical signatures in certain sediment layers, confirming the existence of these recurring fluctuations throughout the Ediacaran duration. Data published in the journal Nature reinforce this view of a period marked by these astral oscillations. Many researchers agree that the Ediacaran, although still lacking organisms with hard skeletons, laid the groundwork for the explosion of life in the Cambrian. Indeed, the additional oxygen allowed for the accumulation of energy reserves, while the first nerve networks paved the way for more elaborate behaviors, such as seeking food or fleeing from danger. Simultaneously, the Milankovitch cycles imposed a rhythm of extinction and diversification, creating windows of opportunity for the best adapted species, and gradually eliminating those that could not cope with these recurring changes. At the intersection of these three factors, the Ediacaran biosphere found conditions conducive to slow but inexorable diversification, preparing the great transition from the pre-Cambrian to the Cambrian. Section 7. For many specialists, the year 2024 marked a turning point in the understanding of the Ediacaran, this geological period just before the Cambrian, more than 550 million years ago. Thanks to new paleontological expeditions conducted around the world and ever more refined analyses, several major discoveries have emerged. More specifically, two new species have attracted considerable attention, Uncus zaugisi and Questio simpsonorum. Their fossils, extraordinarily well preserved, offer invaluable windows into the biology of little-known organisms. Let's begin with one of the most remarkable discoveries, Uncus zaugisi. This name refers to a worm that scientists describe as the oldest known ecdysozoan to date. Ecdysozoans are a vast group that includes arthropods, insects, crustaceans, spiders, and nematodes, roundworms. To simplify, they all possess a cuticle, which is a kind of outer layer they must shed regularly to grow. Until recently, the absence of arthropods and nematodes before the Cambrian posed a serious problem in aligning calculations based on the molecular clock with concrete evidence from the fossil record. Indeed, by comparing the genes of modern animals, it was estimated that the ancestors of arthropods must have existed long before the Cambrian. However, no fossil of this type had been found in Ediacaran sediments, leading to long discussions between paleontologists and molecular biologists. When researchers came across Uncus zaugisi, they certainly did not expect to resolve this long-standing debate in part. Dated to 555 million years ago, this worm is considered a nematode with a cuticle. In other words, it is an animal with an external envelope that it sheds as it grows, much like modern nematodes. Its morphology was found to be unique, a thin and elongated body, devoid of legs, but showing small annulated structures sometimes mistaken for segmental rings. Detailed analyses showed it was not an annelid, like an earthworm for instance, but indeed an echidiosozoan, meaning an animal capable of molting. To reach this conclusion, scientists used highly advanced microscopy methods, combined with three-dimensional digital reconstructions that allow for observing the microscopic details of fossil tissues. The specialists also utilized the organic chemistry trapped in the rock to confirm the presence of fatty acids comparable to those found in modern nematodes. This discovery also brings a welcome reconciliation between molecular phylogenies and fossil observations. When comparing molecular clocks, based on genetic mutations accumulated in different groups, to physical evidence in the ground, it is sometimes difficult to precisely match dates. Molecular biologists had already predicted the existence of primitive arthropods, or their close ecdysozoan cousins, well before the Cambrian, but these creatures did not appear in the sediments. Thanks to Uncus zaugisi, we have a missing piece, a nematode worm with a cuticle, proving that the ecdysozoan group was indeed present earlier than previously thought. The recent discovery causing a stir is called Quaestio simpsonorum. It is a fossil organism that long remained unknown or misidentified until a research team, supported by the Department for Environment and Water, devoted a comprehensive study to its unique morphology. Its name, literally meaning question or enigma, reflects the fact that it displays a question mark shape when viewed from above. The head has a slight curve, while the posterior part curves inward, reminiscent of the punctuation mark. This animal, also dated to the late Ediacaran period, appeared in multiple specimens within a rock layer, known for being rich in atypical fossils. Initially, specialists thought it was a simple assemblage of unclassifiable fossil debris. However, closer observation revealed a coherent morphology, and especially a bilateral arrangement of internal organs, which already implies, at this early stage of evolution, a body organization typical of many current animals, with a right side and a left side. Quaestio simpsonorum's ability to feed while crawling is a key element that allows us to affirm a true anatomical innovation. This organism relies on a general symmetry in which, however, an early functional asymmetry is detected, particularly in the way it moves and feeds. To simplify, one can imagine an animal that possesses a generally bilateral plan, left and right, but uses its right side slightly differently from its left side when feeding. The analysis of fossil movement traces, left all around the individual, indicates that it could gently progress along the seafloor, leaving thin trails. These trails, called crawling traces, suggest that Quaestio simpsonorum might have scraped the surface of the sediment to collect organic particles or fed on microorganisms housed within microbial mats. This apparent functional asymmetry, marked by an uneven use of each side of the body, provides evidence that, as early as the Ediacaran, some animals were already exploring the possibility of using their limbs or organs in different ways. These surprising findings, combined with those of Uncus Zaugisi, overturn the image of Ediacaran creatures being too simple or heavily limited to a single way of life. Although many forms remain enigmatic, it is observed that part of the Ediacaran fauna had already taken significant steps towards dietary specialization, coordinated mobility, and, more remarkably, the expression of functional differences between the right and left parts of the body. It is also anticipated that these organisms had to adapt to a rapidly changing marine environment, with shifts in water chemistry, seabed oxygenation, and the accumulation of sediments carried by rivers. The microbial mats, widespread at the time, played a role similar to a vast field of greenery on which animals could find shelter and food. Quaestio simpsonorum perfectly illustrates how organisms were beginning to exploit this new food reservoir while equipping themselves with structures facilitating their horizontal movement and selective feeding. Section 8. We now delve into the details of the environments that shaped the Ediacaran period. We will focus on three major aspects: basin mapping, eustatic oscillations, i.e. global sea level rises and falls, and the chemistry of certain trace metals. This insight will help us understand how these geographical and chemical factors greatly influence the distribution and early evolution of Ediacaran organisms. Ediacaran basin mapping relies on geological and paleomagnetic studies revealing that large continental blocks, such as Laurentia or Gondwana, were arranged very differently from what we know today. In this ancient puzzle, three regions stand out for their uniqueness and importance in the fossil record: the Avalon carbonate platforms, corresponding mainly to the ancient continent of Laurentia; the Nama passive margins, associated with a part of Gondwana; and finally, the volcanic ridges of the future Chinese platform. Each of these areas was characterized by specific geology, influencing both how sediments were deposited and how living communities were organized along these varied environments. To understand the concept of a carbonate platform, one can imagine it as a vast underwater expanse that is relatively shallow, where the rock is predominantly composed of calcite and dolomite, thus referred to as carbonates. During the Ediacaran period, the Avalon region, attached to Laurentia, offered similar platforms. The generally clear, shallow waters allowed for the precipitation of calcium carbonate and favored the development of microbial mats. These mats adhered to the seabed to extract energy and nutrients. Over time, sediments accumulated in successive layers, resulting in sequences of limestones or dolomites that preserved life imprints. Moreover, the rather modest supply of sediments from continental erosion, referred to as terrigenous sediments, explained the predominance of very pure rock, ideal for preserving fossil remains. Geologists consider these carbonate platforms to have remained stable overall, although they could be periodically emerged or submerged due to changes in sea level, thus modifying the depth of the water on these platforms. On the Gondwana side, the Nama region illustrates a second type of underwater environment: the passive margin. This term refers to the gentle transition between the continent and the abyssal plain, as opposed to an active margin, where subduction or tectonic collision causes strong seismic and volcanic activity. In a passive margin, the continental slope gradually inclines towards the open ocean. The waters there are often warmer due to the tropical or subtropical latitude, which could have been the case in the Nama area. The sediments include both carbonates and coarser deposits, gravels, sands, coming from neighboring continents. During the Ediacaran period, marine temperatures in this region could exceed 20 degrees Celsius, favoring the establishment of rich ecosystems of microbial mats and primitive organisms feeding on these microfilms. The calm conditions of this Nama passive margin, in the absence of strong tectonics, preserved many fossils, offering modern paleontologists the possibility of studying the complexity of Ediacaran populations. The volcanic ridges of the Chinese platform depicted a completely different landscape during the Ediacaran. In these zones, the ocean floor was tearing apart, allowing magma to rise and form an active underwater relief. Seawater, circulating in these faults, potentially became loaded with chemical elements, which were then redistributed into the environment. This volcanic activity could create extreme conditions, both in terms of temperature and chemistry, forcing organisms to adapt or migrate. Yet these ridges could also serve as sources of essential nutrients, dissolved minerals, sometimes crucial for the growth of certain microbial populations or primitive organisms. Beyond the spectacular aspect of potential underwater eruptions, it was the chemical balance of the surrounding ocean that was affected, with inputs of substances that could stimulate biodiversity. The fossils found in these deposits confirm a mix of volcanic and sedimentary influences that highlight the complexity of the Ediacaran ecosystems established on this Chinese platform. Within these distinct basins, the Ediacaran period was continuously shaped by changes in sea level. Specialists refer to eustatic oscillations to describe these global movements of rising and falling sea levels, partly linked to the variation in the amount of water trapped in ice caps, but also to the configuration of the ocean basins themselves. Even though the major glacial phases predating the Ediacaran, such as the Marinoan glaciation, were in the process of ending, more modest oscillations persisted. These could result from the movement of tectonic plates, changes in underwater topography, or various climatic cycles. Observing these oscillations in stratigraphy involves noting where the sea deposited marine sediments over deposits from continental environments, and vice versa. Paleontologists' and geologists' work highlights three major marine phases that succeed one another during the Ediacaran, each associated with a global rise, transgression, or fall, regression, of sea level. During a transgression, waters invade continental shelves, sometimes covering ancient emergent lands and offering new shallow water surfaces. Conversely, a regression exposes areas previously submerged, alters local salinity, and can even isolate marine populations. The amplitude variation recorded for these oscillations is estimated at less than 50 meters, which, on a whole planet, may seem limited. Nevertheless, this depth difference significantly transforms sunlight penetration to the seabed, water pressure, nutrient availability, and temperature, all crucial factors for communities of fixed, bottom-dwelling, or mobile organisms. During transgressions, the influx of fresh waters from the open sea sometimes brought a surplus of vital elements, stimulated biomass production, and fostered the establishment of more diverse ecosystems. However, this influx could also dilute or disrupt certain chemical parameters to the detriment of species highly specialized in a more confined environment. Ediacaran fossils, whether simple impressions of soft organisms or traces of microbial mats, bear the mark of these depth changes. In strata indicating a transgression, species assemblages adapted to more open living conditions are often found, while layers corresponding to a regression contain evidence of more littoral or deltic environments. Plate tectonics plays a background role in this dance of transgressions and regressions. Divergent movements, where plates move apart, create oceanic ridges that can elevate entire regions, thus lowering water depth. Conversely, subsidence due to sediment accumulation or tectonic convergence can deepen a basin, facilitating seawater entry. In the Ediacaran, part of these marine level variations thus stems from the global geodynamic evolution, whose understanding relies on the detailed study of ancient mountain ranges, passive margins, and alignments of underwater volcanoes. Each fluctuation, even seemingly minor, could cause upheavals in community distribution by changing dissolved oxygen availability, nutrient input, and microhabitat distribution. Alongside these geomorphological and eustatic events, the chemistry of Ediacaran marine waters is of great importance. Geologists and geochemists today study the concentration of various trace elements, notably molybdenum and vanadium, to reconstruct the history of oceanic oxygenation and fertilization. Molybdenum and vanadium are termed trace metals because they constitute only a minute fraction of the overall composition of seawater or sediments. Yet, they play a key role. Molybdenum, for example, is involved in the biological fixation of nitrogen, an essential nutrient for most life forms. Vanadium, on the other hand, often reflects specific oxidation or reduction conditions. Thus, geochemists can, by measuring molybdenum and vanadium levels in well-dated rock samples, determine if the waters of that time were lacking in oxygen or, conversely, were sufficiently oxygenated to support life. In this context, we talk about molybdenum and vanadium peaks when we observe a sudden increase in these metals in a specific sediment layer. This rise suggests an external input, usually linked to a phase of hydrothermal emissions. During such a phenomenon, seawater circulates deep through volcanic rock, warms up, becomes enriched with dissolved metals, and then gushes out as hot fluids at the ocean floor. Numerous underwater structures, sometimes called black smokers or white smokers, then release these elements into the water column. The analogy with fertilizer is often used because additional inputs of molybdenum and vanadium can stimulate certain communities of microorganisms, particularly those that require molybdenum to fix nitrogen or other metals for their metabolic processes. In a nutrient-poor environment, these punctual disturbances can trigger mini ecological revolutions, allowing certain groups to spread or diversify. However, an excess of nutrients can also be a double-edged sword. When the environment receives too many nutrients in a short period, the accelerated multiplication of microorganisms can lead to local deoxygenation, as these microorganisms consume the dissolved oxygen during their proliferation. In modern times, this phenomenon is called eutrophication, which often accompanies areas devoid of animal life due to a lack of oxygen. While it is delicate to know precisely to what extent these fertilization episodes were beneficial or harmful to Ediacaran life, in some regions, we observe a coincidence between chemical variations and the appearance of new biological forms. These correlations fuel research aimed at determining how environmental conditions, particularly metal chemistry, may have influenced the emergence of more complex organisms on the long road leading to Cambrian fauna. Thus, we understand the importance of the volcanic ridges of the Chinese platform, where tectonic activity was more intense and could generate these hydrothermal inputs at regular intervals. The submarine floor, traversed by faults, allowed metal-laden fluids to ascend, depositing them in the sediments or dispersing them around. The Avalon carbonate platforms, less subjected to these volcanic intrusions, presented more stable chemical profiles, although they could receive, via ocean currents, elements coming from distant areas. In the case of the passive margins, it is the regular sedimentation that allowed the retention of the signals of these emissions, sometimes for several meters of stacked rocks. Thanks to these stone archives, scientists can today trace the chemical evolution of the Ediacaran Sea, placing each variation in a global context to try to understand its repercussions on local and regional biodiversity. Section 10. At the end of the Ediacaran, a multitude of factors converge to shape an underwater environment in full mutation. We are at the very end of this fascinating era, and oceanic landscapes are transforming at great speed. In shallow waters, communities of organisms once flourishing begin to decline, while new forms, more resistant, attempt to take advantage of this ecological chaos. At the heart of this ultimate stage, the Earth undergoes what are called multiple stresses. This is a conjunction of unfavorable factors that, when acting simultaneously, severely weaken the biosphere. At the end of the Ediacaran, several geological and chemical processes combine to rapidly alter the ocean's balance. One of these fundamental processes is the fluctuation of sea levels. Simply put, sea level is not fixed. It can rise or fall due to various mechanisms, such as the melting and expansion of ice caps or changes in global temperature. At that time, the climate did not remain stable, and it is assumed that significant temperature changes may have accelerated the melting of certain glacier masses in the polar regions of the era. When the sea level rises, coastal areas are inundated, altering habitats near the shore. When it falls, areas formerly marine become terrestrial or lagoon-like. For organisms that depend closely on stable conditions of oxygen, salinity, or nutrients, such variations can be devastating. However, this fluctuation in sea level is just one piece of the puzzle of multiple stresses. Another determining phenomenon is called rapid redox change. The word redox refers to oxidation-reduction chemistry, which involves reactions where certain substances combine with or separate from oxygen. In marine environments, the availability of dissolved oxygen is essential for many organisms, and any significant alteration of its levels causes important physiological stresses. Microbial mats, communities of microorganisms that form sticky carpets on sediments, generally thrive in relatively calm and well-lit waters, where they take advantage of solar energy and particular nutrients. They also contribute to carbon fixation and oxygen release. However, when redox conditions change rapidly, microorganisms cannot adapt quickly enough. They are subjected to sudden fluctuations in oxygen levels, changing concentrations of sulfides or metals, and potential accumulations of toxic compounds. The ever-shifting water chemistry disrupts their nutritional cycles, leading to their retreat or even local disappearance. This sequence of events thus triggers a gradual decline in microbial mats. In the past, these mats largely dominated shallow seabeds, forming vast surfaces that housed a great diversity of microorganisms and small animals, depending on these structures for food or shelter. At this advanced stage of the Ediacaran, the mats are hit hard by rising waters, followed by subsequent recession, and have to cope with waters that are increasingly less conducive to stable growth. The decrease in local oxygen, variable availability of mineral salts, and brief phases of hypersalinity caused by evaporation in coastal lagoons confront these biological communities with extreme conditions. The multiple stresses eventually combine: increased sediment mobility, competition for the few remaining substrates, and the instability of chemical parameters weaken the balance previously maintained by these microbial mats. Result: although microbial mats do not completely and globally disappear, surely some favorable areas remain. A pronounced global decline in their extent and importance in ecosystems is observed. For species that colonize or directly exploit these mats, this habitat reduction represents a serious setback. Indeed, many organisms filter or directly absorb nutrients trapped in the microbial adhesives, while others shelter there to escape currents and predators. Without these mats, the marine environment becomes more vulnerable to erosion, and sterility sets in certain areas, paving the way for new adaptation strategies. All these factors give rise to a phenomenon that can be described as an internal pre-extinction. The term may be surprising, but it reflects the situation. Well before the official boundary marking the end of the Ediacaran, signs of a notable decline in diversity are already discovered. Studies conducted on sedimentary rocks containing fossils indicate that diversity within certain communities decreases by about 20%. To an untrained eye, distinguishing between a thriving ecosystem and one in decline is not always obvious, as fossil preservation is not perfect. However, estimates based on the presence or absence of distinct forms show that this decline is significant. In the past, a greater variety of life forms could be found fixed to the mats or rooted in the soft surfaces. At this stage, many groups that relied on these structures are significantly less abundant. When we speak of carpet-fixed forms, we refer to organisms attached to these microbial layers or deeply anchored in the mud, where the mats provided a nutritional framework. These passive organisms, lacking significant mobility, had to rely on their immediate environment for the supply of organic materials, oxygen, and nutrients. Therefore, the degradation of redox conditions and abrupt changes in sea level primarily threaten them. In contrast, fossil analyses suggest that some tubular or mineralized structure-bearing organisms fare better. By mineralized tubules, we mean creatures whose body wall or certain external parts have solid carbonate deposits. These deposits, although extremely thin at the microscopic scale, can provide extra protection against abrasions, environmental pressures, and perhaps even against some emerging predators. During the Ediacaran, selective pressure is already beginning to intensify. As the environment becomes more chaotic, species with a form of light mineralization find themselves advantaged to withstand sudden changes in temperature, salinity, or even sediment agitation. In certain stratigraphic sections, that is, slices of rock showing the order in which sediments have been deposited, the persistence of these mineralized tubules is observed, whereas softer forms disappear. Everything indicates that by the end of the Ediacaran, life is already experimenting with body reinforcement strategies, which prefigure even more marked diversification at the beginning of the Cambrian. For now, however, these are just the early stages. These mineralized forms do not yet dominate all ecosystems, but they demonstrate particular resilience in environments marked by chemical and climatic volatility. The internal pre-extinction is thus manifested by a gradual transition. Many carpet-fixed organisms, unable to react to rapid environmental upheavals, see their populations decline. On the other hand, a few distant precursors of future compact skeleton lineages begin their slow and discrete rise. Researchers estimate that at this stage, this reduction in diversity is not a simple local event, but a global phenomenon, or at least widespread, in many marine basins of the time. The calculations are based on fossil records from various sites worldwide, sometimes very geographically distant, like in Namibia, Russia, or Australia. The crossover of these data suggests that the average diversity decrease of 20% is approximate, and that there are regions more or less affected. The conjunction of rapid sea level fluctuations, chemical changes in the water, and other environmental disturbances thus contributes to creating a critical situation where one can already sense the imminent end of the Ediacaran. Beyond these figures, perhaps the most striking notion is the disappearance of static forms and the prominence of more robust species. The preferential survival of mineralized tubules provides the most evident proof of this. These creatures likely navigated this unstable phase thanks to their increased resistance and perhaps a rudimentary form of adaptive behavior. Nevertheless, it is important to remember that ecological conditions are constantly changing, and that this phenomenon of internal pre-extinction does not announce the absolute end of all forms of life, but rather a significant reconfiguration of the distribution and abundance of organisms. Simultaneously, with this gradual erosion of carpet-fixed populations, there are revealing clues suggesting a newfound attraction to mobility. The period up to 538 million years ago is rich in discoveries of fossil tracks that attest to horizontal movements on the surface of the sediments. In this context, researchers readily evoke the idea of selection for increased mobility. To understand properly, one must recall the difference between a sessile organism and one capable of moving. A sessile being remains attached to its substrate for most of its existence, whether it's a rock, microbial mat, or sediment fragment. A mobile animal, on the other hand, can crawl, swim, or move, using various structures to escape danger, seek food, or colonize new space. During the Ediacaran period, most known fossils appear predominantly sessile or very weakly mobile. This relative immobility works well as long as resources are abundant and the environment remains stable. However, once environmental parameters begin to fluctuate frequently and intensely, being able to move away from a hostile area becomes a decisive advantage. Indeed, if oxygen is scarce, or if salinity becomes too high in a given area, an organism that cannot move suffers from these unfavorable conditions. In contrast, a living being with sufficient mobility to find a more accommodating microhabitat, even just a few meters away, will have a better chance of survival. At the fossil sites of Nama, located in present-day Namibia, the geological layers from the end of the Ediacaran reveal precisely the presence of three-dimensional crossed tracks. These tracks demonstrate active movement in various directions, sometimes overlapping several layers, suggesting that these animals were able to slightly dig into the sediment or crawl on different levels. Paleontologists study these tracks by carefully observing the shape, depth, width, and orientation of the traces to gather information about the organism's appearance and mode of locomotion. It appears that some individuals could change direction, bypass small obstacles, and follow winding paths in search of food or to escape an area with unfavorable conditions. This adaptive capacity reveals that they possessed rudimentary sensory systems, allowing them to detect chemical or physical variations in their immediate environment. Even if the size of these organisms often remains small, their imprint on the seabed is pronounced enough to be detected several hundreds of millions of years later. Moreover, these fossilized tracks intersect and cross each other, probably illustrating an intensification of biological activity on the substrate. Whereas previously, the sediment was covered by dense and compact microbial mats, a more reworked, more aerated ground is now being discovered because animal mobility stirs and mixes the superficial layers of the mud. This phenomenon is called bioturbation. It corresponds to the totality of disturbances inflicted on sediment by living beings. At this stage, bioturbation remains limited, as many players in the upcoming Cambrian explosion have not yet appeared. Nevertheless, we can already see the signs of a major ecological upheaval. Organisms capable of moving can feed on the remaining mats, expand their living space, and, through their simple activity, reshape the microenvironment around them. This selection for increased mobility is all the more crucial because resources are sometimes scarce and unevenly distributed. The declining microbial mats no longer form a vast continuous layer of potential food and stable support. They rather persist in scattered patches, localized where the chemistry of the water remains temporarily favorable. In such a context, being able to move to find the next nourishing area becomes a competitive advantage. Consequently, lineages that exhibit the beginnings of musculature, or, at the very least, an effective locomotion mechanism, have a better chance of proliferating. Gradually, this dynamic changes the overall composition of marine ecosystems and closely ties survival to a capacity for movement. Keep in mind that we are in the last moments before the official cutoff that marks the end of the Ediacaran and the beginning of the Cambrian, a period which will witness a true explosion of animal diversity. The fossil records of Nama constitute a sort of giant photographic testimony of this moment. Hundreds, if not thousands, of traces representing organisms seeking to escape environmental stress or in search of a better feeding area. These final Ediacaran layers have sometimes been described as a laboratory of nascent mobility, highlighting how the ability to move was about to become a crucial criterion in the subsequent evolutionary radiation. It is not excluded that a form of predation may have begun to develop. The presence of certain perforation marks in thin carbonate shells suggests that organisms were already capable of attacking other species to feed on them. If that is the case, then increased mobility serves a dual purpose. It allows mobile animals to escape oxygen-depleted water pockets, as well as to move away from potential predators. Of course, these early predatory interactions do not yet resemble those that will shape the Cambrian era, where competition will become fierce and defensive innovations much more complex. But they foreshadow the subsequent evolutionary history. From this crucial point, the Earth will never be the same again, as mobility, predation, and rapid diversification will impose increasingly complex ecological patterns, shaping in the long term part of the richness of ecosystems of which we are today the heirs.